Hello, everyone, and welcome to today's live broadcast, Simple Steps to Speed Encapsulated Drug Development. This is Rita Peters, Editorial Director of Pharmaceutical Technology, your moderator for today's event. We're pleased to bring you this educational web seminar presented by Pharmaceutical Technology and sponsored by Capsugel. Capsugel designs, develops, and manufactures a wide range of innovative dosage forms for the biopharmaceutical and consumer health and nutrition industries. The company's unique combination of science, engineering, formulation, and capsule expertise enables its customers to optimize the bioavailability, targeted delivery, and overall performance of their products. Capsugel partners with more than 4,000 customers in over 100 countries to create novel, high-quality, and customized solutions that align with their customers' evolving needs and benefit patients and consumers. For more information, please visit www.capsugel.com. I've got a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit them by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Should you have any technical problems viewing or hearing this presentation, click on the question mark Help widget in the dock at the bottom of your presentation window. Now I'd like to introduce today's speakers. We're joined today by Matt Richardson, Jeffrey Williamson, and Missy Lowry. Matt Richardson is the Manager of Pharmaceutical Business Development at Capsugel. Since joining the company in 2005, he's worked with many pharmaceutical and nutraceutical customers to understand the formulation aspects of their products with two-piece hard capsules. He's been an invited speaker at numerous seminars and webinar topics on hard capsules, cross-linking, capsules for use in delayed release, as well as DPI-based inhalation. Jeff Williamson is Director, Formulation and Pre-formulation Department at Capsugel. Jeff and his team are responsible for drug substance characterization, drug product formulation, and process development. Missy Lowry is the Senior Marketing Manager, Americas Region at Capsugel, and serves a diverse customer base in health and nutrition, pharmaceutical, and over-the-counter products. Thank you all for being here today, and Matt, you have the floor.
Oh, thank you, Rita. So first, let me give you guys a brief overview of Capsugel's technology platforms. So Capsugel is a specialized partner to pharmaceutical clients with focus on addressing bioavailability challenges and modulating PK for challenging compounds. Our approach is pretty much an unbiased in that we fit the best technology to the problem statement. Target product profile, commercial objectives, they're all big parts of what we ask and what we work with customers to achieve. Our depth in technologies allow us not to push any one technology or its boundaries or even finished dose formats. So we try to work with customers to optimize the formulation, increase its chances of therapeutic success, and minimize reformulation work later in development. All important aspects in bringing good customer service and product development simultaneously. So as such, we have several different key areas. And that's very different. Capsugel has always been known as the hard capsule manufacturing plant. But today you can see with this slide, there are so many other platforms that are available. So we actually have depth in several different key areas. That first area, what we'll call the first pillar, is bioavailability enhancement. It addresses dissolution rate and solubility challenges using micronization, nanomilling, amorphous dispersions, and lipid-based approaches using various liquid-type operations as well. The second, what we'll call the pillar there in the orange category, is what we call targeted delivery. So we utilize an array of technologies to target specific areas of the gut or modulate dissolution profiles as required. And increasingly, we have projects that require both solubilization and PK modulation at the same time. Of course, we're known for our capsule and encapsulation technologies and offer a range of specialized fit-for-purpose products and approaches. And in this section of the webinar, I want to introduce various ways to think about streamlining the research and development activities through the strategic use of these types of capsules and their applications. We know that a lot of research into new molecules begins in capsules and then moves away as various challenges are introduced into development. Today, however, with a little planning and some ingenuity, the use of capsules can be applied to a larger array of molecules than ever before. We'll start off the topic with a discussion of polymer choice, which remains an oft-asked question. Most customers know the route of administration fairly early on, and that knowledge is probably the first line to follow when choosing the best polymer for the situation. For instance, if we know that the pulmonary route is the best to deliver a treatment and we consider capsules for inhalants, then we know that we need a polymer that's robust enough to work in a device that either pierces or cuts the capsule to access the medicine. We take then into consideration the formulation and or the API properties that are really essential to protect the product from the physicochemical factors that could challenge the formulation downstream. Mindful of both of these, we can then choose a capsule polymer that is best attuned to the formulation for successful product development, and then equally as important, suggest a host of complementary tools and development strategies to further streamline activities. The choice of the right capsule polymer is extremely helpful in streamlining development. I usually sum up the idea in the simple thought of choosing the right capsule polymer means that I don't have to go back, start over, and repeat initial activities again and again. So in this chart, you can see a few very important capsules that are essential for early R&D studies, including quite new enteric HPMC polymer that I'm going to spend some time discussing in more detail later on in the presentation. Included in the chart are several of the most important physicochemical factors that correspond to each one of the capsules as well, and these factors are almost always considered when choosing the right polymer. Consider, for instance, the desired release profile. Is it essential for the product to be immediate release, or is it a molecule that is going to be sensitive to acidic pH, or perhaps it's a moisture-sensitive molecule? Whatever the critical nature of the product that you're working with, the right choice of the polymer can go a long way to really protect the product. For immediate release applications, we've seen a large and a growing interest in the VCAP Plus HPMC capsule, as it offers multiple ways to protect a formulation and sometimes even enhance its performance. So I want to spend some time highlighting why that's the case. One major aspect to consider is what I call the window of opportunity or the window of success with respect to moisture. Gelatin capsules have always been robust in our industry for a multitude of different purposes. But we recognize and have always advised that they be kept within a recommended storage window of 35 to 65% relative humidity to ensure that the gelatin remains at 12 to 16% water content. And that's going to be the optimal performance window for gelatin. Straying too far above that level and brittleness, below that level, brittleness starts to come into play. And if you go too far, it actually leads to manufacturing issues as capsules start to deform slightly, lose their shape, and don't perform well in the actual encapsulation equipment. As VCAP Plus capsules are made of HPMC, they don't rely on water content for pliability. And that window of opportunity is actually larger than that of gelatin. And that's represented on the graph on the right side. HPMC capsules can go as low as 2.5% to 3% before capsule brittleness is even introduced, and even then at an extremely low level. As importantly, HPMC capsules contain 2 to 9% water across that range, and that's an average 6% less water than you'll find with gelatin capsules. In early studies, just how hygroscopic or moisture sensitive a molecule is may not always be well defined. So starting with a capsule that better levels the playing field is an excellent idea. And add that to removing concern of capsule brittleness and having less concern of using desiccants for product protection, make it a really important feature for early formulation work. But when considering the use of HPMC capsules, it's going to be important to realize that not all HPMC capsules are going to be an equivalent change from a gelatin capsule. So HPMC capsules can be made by two very different manufacturing processes, one which utilizes a gelling system and one without. The concern with the use of a gelling system is that inevitably some variability is going to be introduced. And this slide is very good at revealing just how much variability can be seen in vitro. So if we dissect this a little bit, and I'll show you the graph, you can see there's a pretty big difference between ionic strengths and how that affects the dissolution character. You can see a wide variation then when you go from pH 1.2 by the USP to the pH 6.8 by the JP2. There's a little bit there, and changing that they both have low ionic activity. But then when you look at pH 6.8 by the USP, which is considerably more ionic activity within it, you can see a large variability. And as you increase the amount of potassium chloride, which affects the ionic activity, you can see even more variability introduced. And the same exists when we use simulated milk fluid there in the purple versus that of pH 1.2 to represent both the fed and fasted states. And it seems like that would not be, that's something artificial, but consider that just drinking a glass of orange juice or eating a few strawberries or even taking a vitamin along with your medication can all affect that amount of variability for a product. Then it really hits home how quickly this can come into play. If, however, we remove that gelling system then, and that's another essential point for the VCAP Plus capsule, is that it contains no gelling system, and it gives you a very different result as far as variability goes. VCAP Plus capsules are made by a thermodylation process instead of a traditional HPMC manufacturing process. And without the gelling system afforded by that traditional manufacturing process, the level of variability that you see was just greatly reduced, and I think that's represented well on this slide. If you examine the exact same in vitro dissolution protocol shown in the previous slide as in this one, we see that the lack of gelling system greatly reduces the previously shown variability. And no longer can you find any variability and draw significant conclusions between changes in pH, ionic strength, even fed and fasted states. One area, though, that remains in question for some customers, though, is that apparent lag time in the initial dissolution and disintegration of HPMC. And you can see it on this slide. You can see up to 5 to 6 minutes. It doesn't appear that dissolution has actually set in. And so we often get a lot of questions about that from customers. So to further address any concern with that apparent lag time, the VCAP Plus capsule, and to better understand the performance of capsule overall, we commissioned an in vivo study to compare the performance of the VCAP Plus HPMC capsule made by thermogylation, and then compared it against the gelatin coning snap product. In the study, we're looking at Excedrin extra strength caplets, and we chose those because Excedrin has 3 different biomarkers within it, and each representative of a different BCS classification. So we took the Excedrin extra strength caplet and we added it to both a VCAP Plus HPMC capsule and also to a hard gelatin capsule. The capsules were dosed according to the protocol shown, a randomized 2-way crossover study, and the various PK parameters collected as part of that study. In the results from the acetaminophen marker, it's clear here, it's clearly shown that neither the difference in the polymer type nor the slight lag time in HPMC disintegration affected the performance of the product in vivo. Here, gelatin is represented in blue, while the HPMC VCAP Plus is represented in red on the graph. And it's seen that the performance is nearly identical with high confidence intervals being represented. Take a look at the second biomarker, and the same can be said for the acetyl salicylic acid marker as well. Again, the gelatin is seen in blue, and VCAP Plus HPMC is found in red on the graph. When the values are examined, the data table below the biggest difference was found in the intra-subject variability. But otherwise, the product performed very closely regardless of the polymer of the capsule, and this time with a molecule from a separate BCS classification. And then we can compare the third biomarker, which was caffeine. Even here, with a BCS class 1 molecule in which fast dissolution is characteristic, the roughly 5-minute lag time performance of the VCAP Plus HPMC capsule in vitro shows no bearing in the overall in vivo performance between the two different polymers, again with very high confidence intervals. So hopefully with this in vivo study that we show here, we've demonstrated that HPMC capsules can really broaden what we would consider the working quality by design space for hard capsules, provided that the HPMC capsule are ones manufactured by thermogelation, which contain no gelling system within the capsule. The lack of the gelling system greatly reduces variability seen when traditional HPMC capsules are used. So adding the in vivo data study to the previously shown physicochemical benefits of the VCAP Plus capsules, it really becomes easy to see why these capsules are a great choice for the right first time formulation work, and greatly reduces trial and error efforts. We see a lot of momentum in customers working with new chemical entities starting out in VCAP Plus HPMC capsules today, if not in place of hard gelatin, at least in parallel studies with them. However, there are a lot of formulations which it's desirable for the formulation to be delivered past the low pH of the gastric environment due to either acid sensitivity or even sometimes when application of delayed release is beneficial. Until just recently, doing this through the use of capsules has required some sort of additional coating process to be utilized on either the formulation contents, like the different multi-particulates, or even on the capsule itself. We're excited to announce recently the introduction to the new VCAP enteric capsule to the portfolio of products that we offer. The objective of the creation of the VCAP enteric capsule was to offer a quicker way to evaluate products that need enteric protection without going through an extensive, time-consuming process to accomplish that goal. The enteric properties of the capsule reside within the polymer itself, not an exterior coating on the capsule, and they perform according to the requirements of all major pharmacopoeia without any additional processes that are typically needed after filling with the formulation. The VCAP enteric capsules are two-piece, hard capsules which conform to standard size for all powder fill capsules. They are made by a thermogelation process from a combination of HPMC and enteric cellulosic materials which have been used within the pharmaceutical for over 20 years. So there's no concern of introducing a new or untested material. You can see the performance of the capsule in the graph during the two-stage test protocol that's applied. Compliant with all major pharmacopoeia, less than 10% release of the product is found after 2 hours at a gastric pH, while a quick release is achieved when the capsule is added to the higher pH buffer of the second stage. It's really important to note here that the VCAP enteric capsule doesn't require an additional sealing or banding step either. And that, in order to get it even compliant, to make sure that it's compliant with pharmaceutical requirements, we would typically need to either band or seal the capsule. But this particular VCAP enteric that we're just introducing, it requires no additional processing from either a sealing, a banding, or a coating process later on. So you've got an effective enteric that you can add to your protocol early on with minimal processing steps. And that really does add to the power that a formulator has today to choose and look at early stage work. So having non-complex enteric options at early drug development can really open a lot of opportunities for rapid advancement of what we can learn about a new pharmaceutical and then its subsequent development from there. So in this slide, we did a study to show how powerful this kind of tool really can be. Entecort was chosen as a baseline compound as it is delivered enterically by the use of coating of microgranules to achieve acid protection. To explore the performance of the capsules, budesonide was dosed as pure API directly into the VCAP enteric capsule. The two-stage dissolution protocol was used to compare the performance of the brand product versus that of the VCAP enteric capsule that's loaded with budesonide. And you can see from the graph that both routes provide excellent enteric protection and then open quickly at a higher pH stage, offering very similar release profiles for the products, without going through that formulation and additional coating steps for the microgranules. And that gives formulators a bigger advantage than ever. So as a reminder, the VCAP enteric capsule, they're not the only enteric capsules within the Capsugel portfolio of products. It adds to the intrinsic drug delivery system already established within the product line, and we've been demonstrated over the past couple years to provide complete enteric protection from the acidic environment. So customers have a choice of the enteric offerings that you have, and choosing between them is a decision of what's best both for the API combined with a preferred development route. Either choice offers excellent enteric protection and provides traditional benefits of stomach protection from harsh APIs and delayed release opportunities as well. The intrinsic drug delivery system differs from the VCAP enteric capsule in a few different but yet important ways. While also utilizing capsule technology, the intrinsic capsule is a drug delivery technology which is available for license. And typically this is a project that we would partner with a company but undertake in-house at Capsugel to perform. Another large difference in the intrinsic is that it guarantees 0% release in the acidic media. The VCAP enteric capsule is a ready-to-use capsule that can be sent directly to a customer for evaluation. It ensures compliance with all pharmacopeal standards of no more than 10% release in acidic conditions. And therefore, it differs just a little bit more in that versus the intrinsic drug delivery I just spoke of. Some molecules are going to be able to operate well within that standard, but some are going to require that 100% acid protection of the intrinsic capsule. And so deciding between the two, again, it's really a choice of, do I want to work with something off the shelf, or would I want to go through a drug delivery system? And what's good for the molecule? Do I need a 10% or less, or do I really need to go for absolutely no release in the gastric environment? Okay, so for past formulation studies then, there are capsules meant to assist with streamlining the clinical phase of product development as well. These capsules are called DB caps, and the DB refers to the double-blind clinical trials that have become so prevalent today. DB cap capsules have a unique shape compared to traditional capsule used for solid dose encapsulation. The body and the cap have been designed with a shorter length and a wider diameter. And there's a twofold benefit to the design in that the capsule was made shorter so as to be easier to swallow, while the increased width can actually accommodate greater than 90% of the marketed solid oral dosage forms that are on the market today. The cap has been designed to fit much longer over the body of the capsule, making it difficult to get into the capsule to break the blind, and nearly impossible to do so without some visual evidence of tampering. The globally acceptable colorants used in the capsule shell allow for a complete blinding of product contained inside and allows for a wide range of oral dosage forms to be compared quite easily. And this would eliminate the need to have identical placebo matching products made and negates costly labor-intensive operations like de-embossing or other identification removal processes. As a complement to the DB caps, there's a sizing guide that allows for rapid selection of the most appropriate size of DB caps needed for the project. They come in 8 different sizes, and most commonly used DB caps are available in either gelatin or the VCAP Plus polymer to accommodate any formulation challenges that might be required as well. For proof of concept studies and early animal testing, another strategy is there's a capsule that is available as well, and we call those PC capsules, with the PC in deference to the preclinical studies in which they are used most often. PC capsules allow for encapsulated formulation to be dosed for rodent studies. These are size 9 capsules by comparison to standard powder-filled types, and roughly 10 to 15 can easily fit on a small coin. So in the upper right side of the slide, you can actually see how the size compares to even a standard paper clip. These capsules are used typically with API alone to dose the animal so as to bypass formulation time and to get proof of concept results quickly and efficiently. In addition to API and capsule, though, they've actually been used to house a variety of formulation types, such as semi-solids, liquids, in addition to the powders, and the added benefit of taste and odor masking certainly help with the rodent study. They can be coated to achieve enteric or targeted release if desired as well, and that makes them a powerful tool in the rapid advancement of early formulation work. Lastly, I'll offer up the use of the all-color capsule as another strategy to advance development of new products. The concept of the all-color capsule is revealed in its name. It's a capsule that contains multiple colorants and is available in 2 different types, one containing only global colorants and the other containing the most common FD&C colorants that are accepted by the major regulatory agencies. The power of the all-color capsule is the strategy of the ability to move forward with stability studies in the absence of a final choice of color and ink that's often provided by marketing. Quite often, the final commercial design is yet to be determined when it's time for stability studies to begin. The all-color capsule allows for these studies to proceed in such of a situation as that, where the formulation is exposed to the dyes during stability and simply dropped from the final formula when the color has been chosen. And regulatory-wise, the lowering or removal of dyes is a perfectly acceptable process. The all-color capsule comes in 2 different varieties, as we said. Here's the one that is created from the use of only global colorants, and so that will be the iron oxide trio as well as titanium dioxide and blue 2, and you can see the different color extractions that can come from that all-color capsule versus that of the one that is made from FD&C type colorants as well. And these FD&C type colorants are the primary ones that are used and widely used in major pharmacopoeia, but yet not quite global. So you'll see a lot of applicability in the US, Europe, and even in Japan with some of these, but not utilizing all the different FD&C dyes, as each country has a completely different palette of acceptability for these different things. And so you can see how the strategy of the all-color capsule comes into play to really speed development as well. And with that, I want to conclude this section, and I'm going to turn the presentation over to my colleague, Jeff, to pick it up from there.
Thank you, Matt. Good morning, everyone. For fast-track formulation and development technologies, just wanted to, as far as the support of the capsule programs that has been recently described from Matt, wanted to talk a little bit about microdosing, API and capsule. Here at our Tampa facility, we do a API and capsule as a first approach for streamlining the clinical reads for certain APIs. So simply put, what is API in a capsule? Basically a powder precisely weighed into the capsules as been previously shown. Doses as low as 100 micrograms can be accurately dispensed with an RSD of typically less than 2%. What you see here is an image of microcrystalline cellulose in a size 3 capsule to give you an idea of what an actual fill volume would be for that particular presentation. So basically, how is it done? Drug substances or APIs that are brought into for evaluation are looked at from a particle size distribution, flow, fill weight volume. We certainly do any type of physical assessments on the drug substance or API to be introduced into the capsule. These are the types of devices that are currently used, Accel-Dose 120S and the Accel-Dose 600S. The 120S is used primarily for preclinical evaluations, small-scale evaluations, that we can get some initial reads on how the powder actually performs during this type of operation. This operation is not a tamping mechanism. It's delivered by a tapping or delivery system, whereas really a key attribute is flow characteristics. Moving forward into clinical trial manufacture and small-scale runs, we can use the same types of principles here in developing these dosage forms. So benefits for API and capsule: streamlining product development, increasing speed to clinic proof of concept. So using some of the capsule presentations that were previously described, we can do direct applications at our Tampa facility where we introduce the API into whether it be an all-color capsule or an enteric capsule to do initial evaluations. Minimizing the drug substance usage. The precision weighing activity uses very little API for early evaluations. So if API amounts are critical or are limited supply very early on, the reduction in waste from this particular process is necessary. We also have the ability with the API and capsule to eliminate any excipient compatibility screening. Basically looking at the drug substance or the API itself inside the capsule shell. Simplifying analytical and stability evaluations, because there's basically no other excipients in the formulation or the drug product here that would slow the actual process down. Again, just to add the programmable and precise dispensing of the drug substance into the capsules, each one of these capsules that are produced on the Acceladose systems are accurately weighed, and that information can be acquired for each one that's actually produced. So for minimizing analytical and formulation development costs, basically streamlining by adding the drug substance or API into the capsule shell, whereas your analytical endeavors are not challenged with excipient compatibility or looking at formulated dosage forms, you have API and capsule shell. And this next slide is a case study for pediatric capsules. One of the challenges that we had here is a pediatric dosage form that was needed for sprinkling on a food substance to administer to pediatrics. The 2-pellet fill system that we were employing required a very low dose, 7 to 8 pellets. One of the biggest constraints in using something as so low would be the entrapment of the actual pellet in the sachet. We were able to demonstrate on the Acceladose using the sprinkle cap that we could administer the pellet fill on the Acceladose as a viable option instead of using the sachet. Additional capabilities for fast-track development we used here. Again, for lipid-based formulations, we use a CFS 1200. In the CFS 1200 and the Caps presentation, we're able to introduce liquids into capsule presentations where no banding is really required. It uses a LIM system, which is actually a microspray sealing system, and you're able to deliver formulated products from 20 degrees to 70 degrees Celsius using this particular system, whether it be in an oil format, an oil formulation, or a semi-solid formulation. We're able to introduce those into the Caps capsules. Also in development, we have a FlexiLab system where we can introduce 2 powders in a filling operation, use about a powder or a bead filling operation, or even 2 beads, a multi-particulate presentation in early development. Powder micronization. One of the first approaches with APIs and looking at enhancing bioavailability is simply to micronize the drug substance. As many of you are aware that the drug substances in very early stages can come in a variety of different particle sizes, and some of the challenges are inconsistencies with lot-to-lot production. We also have the ability to micronize or mill the APIs to get a consistent particle size distribution from the development throughout the process. So in conclusion, this gives you an overview of some of the capabilities that we have here at Capsugel Tampa. At this point, I'd like to turn it over to my colleague, Missy Lowry.
Thank you so much, Jeff. I appreciate that. I'm going to be talking about a few of the R&D solutions that we've developed. These are going to be tools and also processes and approaches to help you streamline and achieve your R&D results in a quicker time frame. The first of those R&D solutions are really brand and color development shortcuts, and a lot of that has been created as a result of the new VCAP Plus product. In order to ensure that VCAP Plus is available in the color that you need, we've developed more than 200 colors, pre-formulated them and validated them in manufacturing, and identified what the global pharmaceutical acceptance is for the color options. And that particular tool is available on the Capsugel website or from your territory sales manager. Along with that, we've also developed what we call print rolls, and these print rolls are for your use when you're preparing your R&D stabilization batches. This allows you to use a print roll to imprint the capsule for stability, and no development is required for the print roll to be used. As you can see, we can print axial, radial, or a combination of axial and radial print together. I do a lot of work in the area of brand identity, working with customers who are interested in optimizing the patient experience with their particular drug. And I've put together a consulting approach, free of charge, on understanding the impact of color on patient compliance. And I think that this is something I put in just a few slides to kind of give you a sense of what you can help to accomplish utilizing the right psychology of color. We know that capsules are well known and they're a well accepted dosage form. What's important is that the color helps to not only build the brand. As you can see, Prevacid and Nexium are two examples of well-known products that are identified by their color. But color also helps the patient to be able to have a better experience with it as well. Color can really impact proper dosing. We know it helps avoid mix-ups among the elderly. They're able to identify what they're taking. And in the research that we've done, it's actually shown that color is probably the strongest aid in drug identification, even more so than the name or the dosage form, the shape, or the size. Our research has also shown that patients prefer capsules, and they prefer them because of their ease of swallowability and their shape. And because they're able to mask taste and odor, and they have that smooth, glossy shell and that rounded, narrow shape, they physically and visually look like they're easy to swallow. And our research has backed that up. So when I work with a consultation on color, these are the questions that I ask. What's the therapeutic class of the drug, and which effects are expected? What is the targeted patient population? Is it going to be pediatric, or will it be for those over the age of 65? What other medications already exist in this therapeutic class, and with which products will this medication be combined? Clearly to see if there's a color that we need to avoid in order to avoid mix-ups. And then, will the product be marketed in different dosage strengths? Do we need to look at families of colors as an example? And where in the world will the product be marketed? As Matt mentioned, there are different regulations depending on what part of the world the drug will be distributed. And finally, will the product have a pediatric indication? Do we need to incorporate that and be concerned about some of the color regulations that exist in certain countries related to pediatrics. Patient acceptance is an important psychological consideration because patients recognize their medication by the color. And they also know if their brand has been switched, if that color is not close to what they expect to see. There really are a whole series of rules that exist for associating colors with indications, and this research in many different reports is also on the Capsugel website. But as a part of our overall consultation, we generally pull together the type of research that might impact a color choice based on a particular classification or therapeutic indication. And sometimes it's not what the patient cohort accepts, but it's what the patient cohort rejects that's most important. An entire field exists for this emotional response of colors and has been used quite often in the pharmaceutical industry. And I know that given the dozens of consultations I do on a regular basis, it's important to take that into consideration. This is just one of the studies that was done. Dr. Huskisson out of the United Kingdom did a study on the analgesic effect of different colored capsules. And they were placebo capsules. And as you can see, the red had the highest score for pain relief, even though the capsules were identical. Where that comes into play, thinking about it, if you were to apply that to fibromyalgia as an example, a white-red or a yellow-red combination would be both easy to distinguish and carry a real analgesic effect. And there's an example of, in that analgesic category, a customer who took that recommendation. So color choices are a complex opportunity. The patient with the consumer emotional impact, the regulatory environment in which the drug will be distributed, and the market position that you want to take. Choosing that right color was our objective when we developed a novel solution. Besides our consultation, you can build your own capsule. And this new capsule tool is on the Capsugel website. It allows you to make optimal color decisions, and it provides a guidance for regulatory by each of the colors that have been pre-formulated. This will help you be time and cost effective, and formulators, regulatory specialists, and markets can move more easily to market. I'm going to show you that in just a moment. You access the app from the Capsugel website. It's called Build Your Own Capsule. You actually can request a color that you develop directly to customer service. Customer service will develop that formulation, provide you with a color card, and develop a quote in process. So showing you how it works on the Capsugel website, build your own capsule in 5 easy steps. It starts with generating a color request. And what you'll do is you'll go in and you will select your design, and then once you've developed the capsule, you'll actually send it directly to a CSR. And the CSR can take that up as if they were developing a color request that you had made via a telephone call. So here's a view of how it's going to work. So you follow these steps. First, select your capsule type. We have many polymers out there, but they're all represented with the colors that work best with that particular polymer. You select the size and then the target market. Are you pharmaceutical or are you health and nutrition? Both are represented separately because of the regulatory environment. Once you do that, you can select a color combination, and then you can also define how opaque will the capsule be. You select your colors, and the different options are open, which allow you in 3 dimensions to view the capsule itself as it's being built. And at the bottom, regulatory information is highlighted. On the Build Your Own Capsule, you can also choose printing options and then save your design. Use that in a presentation to colleagues or just to view your capsule history. And, as I said before, you can also request the color sample based on your design. So it's a simple but powerful tool with regulatory overlay designed to help you speed to market in your decision-making process. So for color and print, we can help you. We can advise you about patient response to color. We can also help you with the regulatory understanding. This new web-based tool helps you as a formulator, a scientist, or a marketeer to be able to determine what choices you would like to make. And the feasibility of those colors and validated colorants can help speed you along in the process of creating what you need. We provide R&D samples in a variety of different ways to you, and many of them are at no cost in order to ensure that you've got what you need in your laboratory as you're developing it. Marketing samples are free of charge and are available in all of our polymers and all of our sizes, but you can also order R&D bottles of them, and they're ready to ship within 48 hours. There are about 2,000 capsules per bottle, depending on the size, and they too are free of charge. If you have a small project that you want to run, instead of buying an entire carton, you can get what we call a capsule caddy. And that capsule caddy size, color, and type of capsule is available on a simple brochure that we've got called our R&D offer and can be requested. You can request as a result of this webinar or from your territory sales manager. And then finally, we create custom R&D orders, and we take very close attention and pay extraordinary attention to the timeline as we develop an R&D order, because we know you're in a very fast track. So our R&D orders are available starting from the choice of color to the final shipment within a 3-week timeline. And we're now extending that into the VCAP Plus as well. I'm going to go back one. I just skipped through one. We just introduced new fiber-free packaging as well. What this does is this provides a cost-effective solution for transporting and packaging empty capsules within a cGMP environment. And your R&D orders will come in this type of a fiber-free configuration. It's a polypropylene system of both carton and pallet, which is really what the leading-edge pharmaceutical environment organizations are starting to practice as a way to reduce risk and speed production. The real benefits of fiber-free are that they meet the cGMP requirements associated with internal quality systems. They're both environmentally responsible and they maintain robust traceability by avoiding any kind of internal transfer within the manufacturing environment. We also have handheld systems and tabletop systems that are available to you. There's more information on the Capsugel website related to these, either the PC Cap kit, the multi-filler kit, or the 100-hole Pro Filler or the 300-hole Pro Filler, which provide a cGMP acceptable tabletop filling system that can go as high as 9,000 capsules an hour. For R&D environment, we've also developed an Ultra 3, which is a high-speed semi-automatic filling machine for up to 30,000 capsules per hour with an auger filling system. This, too, is helped to design to speed you to market, to be able to address the needs of your R&D laboratory and also assist you in your formulation process. So with that, Rita, I'm going to pass it back to you. Thank you very much.
Well, thank you very much, Missy, and also Matt and Jeff for those presentations. Just a reminder to the audience to how to submit your questions. You can type them in the Q&A box, which can be found on the right-hand side of the presentation window. And we'll try to get to as many questions as we can. Missy, actually, we're going to start off with you. For the capsule color printing, is titanium dioxide used for the ink? And if yes, do you have an alternative for companies where that material is prohibited?
For the ink itself, it would depend on what color is required. If it's for the capsule, titanium dioxide is most commonly used as an opacifying agent within it. So if opacity is not a consideration, it can be excluded from the color formulation. In terms of best opacifying agents, TiO2 is probably the most commonly used, And what it does provide is excellent UV protection for the formulation inside.
Okay, thanks. Matt, this one's for you. Do enteric capsules need to be banded to maintain their enteric properties? And can you completely skip the step of having to enteric coat by using this product?
Okay, so both the VCAP enteric capsule and the intrinsic capsule, to my knowledge, do not need to be banded or sealed in order to get the full enteric protection of the product. And then because the enteric nature is actually intrinsically in the polymer itself, there is no need at all for additional processing steps later to coat the capsule to really perform the enteric. Now, I can only obviously speak to the capsules that we spoke of today, and in the Capsugel pipeline, I understand there are some others that are on the market, and I understand that they do need banding. So, but to be specific, the VCAP enteric and the intrinsic drug delivery system that we spoke of today should not need those.
Okay, thanks. Jeff, we're using API in a capsule. Are there many real-world examples where taking this step has reduced product development time by several months?
Yes, there are. We have an example recently where we had a program that actually entered the clinic that started in parallel as an API and capsule and also a formulated product, whereas our formulated product has progressed from excipient compatibility into basically dosage form assessments. The API and capsule has already been introduced in the clinic.
Okay, great. Missy, this one's for you. I'm interested in the all-color capsule to help speed up our R&D work, since marketing usually takes extra time to decide on color. Is the best process to go ahead and file with the all-color capsule?
It's one that we have more customers doing every day because what you've got is the ability to be able to show marketing the two different, I call them extraction capsule cases, which gives them a good indication of the different types of colors that they can choose from for their final finished marketing form. And what we found is that most marketing groups go through a thought process that allows them to be able to say, yes, well, it's going to be either global or it will be an FD&C combination, and this would cover as many of the different colors we might choose in the final form. And it also allows an extraction of the inks on the outside as well. So it covers all colors and it covers all ink colors as well.
Okay, great. Thank you. Matt, can the PC cap filling kit be used for OO capsules for dogs?
Actually, that's a really good question, and I'm assuming 00, and I don't know right off the top of my head. It will be something that I'll have to investigate. So I will do
right,
that.
I was going to say that actually the PC cap is designed for rats and guinea pigs. So I'm not certain, given the size of that it would be useful for dog studies.
All right. Well, we can follow up on get a response to the person who requested after the webcast. Matt, another one for you. Can we get enteric DB capsules?
That's a good question. The answer is today, no. In the future, I think that you will be able to get the BCAP enteric and probably also a PC cap and probably a DB we will look at as well. Because the VCAP enteric is very newly introduced to the market, we're actually rolling it out as of CPHI and AAPS this year. Today, the design is limited just to the powder Connie Snap type.
All right. Here's our next question. Jeff, what are technologies available at Capsugel to test and investigate for cross-linking in gelatin capsules?
For testing for the cross-linking, mostly would probably be from your analytical responses. Specifically, if
heads down the degradation pathway or you see some slower dissolution rates.
Okay, Missy, how do we go about requesting a sample bottle or a capsule caddy for testing purposes?
It's easy to do it, and it really just requires you to get in touch with us. You can send me an email if you'd like. You can contact your customer service representative if you're one of our current customers. You can attach that question to the bottom of this, to the Q&A portion of this webinar. What we do have is, as I said, is a full R&D portfolio that has a lot of useful tools within it, including a listing of all of the sample products that we have, the sizes of all of our containers and how many capsules are in each of them. It has also the VCAP color tool as well as the print tool and other information about technologies used in the R&D lab. So contact us. You can contact us also through the Contact Us screen at the end of the Capsugel website, and that comes directly into our offices.
Okay, great. Matt, for you, we've typically used gelatin capsules in the past for our new R&D developments. You mentioned some benefits of using different polymers. So does your team help customers go through the pros and cons of choosing polymers for their formulations?
Absolutely, we do all the time. I'll be happy to do that individually with whatever customer. There is a series of, there's probably different things that we would go through, but we basically start with an evaluation of the molecule. What kind of properties does it look like? What's your application look like? And then from there, we use a series of targeted questions to really hone in on what's going to ultimately be the best polymer to work out from a physicochemical standpoint.
All right, Missy, you talked about different types of color research. So if someone wants your help in choosing a color, what do they need to do?
Again, go ahead and contact us. The other thing I would recommend is go to the Capsugel website, to the Knowledge Center, and a good number of these studies are residing there. But contact your CSR, or you can send me an email directly, and we would be happy to start that conversation with you. There's no charge involved in doing this. What we want to do is help you to create the most successful color choice for both the therapeutic category as well as the regulatory environment in which you need to distribute your drug.
Okay, Matt, are there any enteric capsules for liquid or semi-solid products?
Not today. So again, we have a design for a liquid and semi-solid capsule called the VCAP, and I expect that that will be a future way that we take the VCAP enteric program. But as of today, no. We would work with you to, there are other ways that we would do it, but ultimately I believe that we'll get that VCAP enteric on the market.
All right, we have one final question, and that's for Matt. How do you minimize delays in dissolution of over-encapsulated products?
Okay, that's a good question. So for the most part, it depends on the kind of molecule that you're working with. Are you working with something that's really fast, immediate release? What you'll find is that typically there's been several case studies and some marketed products and also, I guess, some published literature that show there isn't any significant in vivo difference in an over-encapsulated product, whether it be gelatin or I believe in HPMC as well. So in the case that you are seeing that, we want to take a look. Is it because something's going on, or is it really because of an over-encapsulation situation? And then we would address it in a couple of different ways, sometimes even by adding a different type of backfill that helps speed up either water solubilization or even a disintegrant can help in that mix as well. But it's all case by case. So we'd be happy to work with you guys on trying to diagnose exactly what the problem is and then figuring out a strategy to improve it from there.
All right. Well, thank you very much. Unfortunately, we are out of time. I do want to thank our speakers for their presentations and the audience for listening in and submitting their questions. And also I'd like to thank Capsugel for making today's webcast possible. Today's program will be available for on-demand viewing through November of 2017. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. We invite you to forward that announcement to any colleagues who may have missed today's live event. In addition, audience members will receive a white paper summary of this presentation. Will be made available in a few weeks. Finally, invite audience members to take the time to complete our sponsor's brief survey once the webinar concludes. So thank you for joining us today. I'll see you next time, and have a great day.